Hi, hello. So as Perry mentioned, from South America, and I have all these evil strains you are talking about earlier today. So this is a work that I am developing with Zhenyang Lu. She's in the Australian National University. And we are working to use comparative genomics to decipher the host adaptation of Austropuccinia psidii in two host species in the Myrtaceae family. Okay, so this is a collaborative work between Brazil and Australia. So as you may see in this slide, we have several universities involved. So I am from the University of São Paulo in Brazil, but I had spent the last 4 months here in the Australian University working with the Schlesinger Group. Perry Tobias and some other collaborators of the University of Sydney, also the University of Western Australia, and some collaborative work from all over the world. Okay, so this was already mentioned today, but I want to bring back the world perspective of Austropuccinia psidii. So we have these two lineages, the South African biotype and the pandemic biotype, that are polyphagous, infecting hundreds of different hosts, and represent a biggest biosecurity threat, and also brings economic impact for all those regions where it's present. On the other hand, in the center of origin of Austropuccinia psidii, the pandemic biotype was only detected in Colombia thus far, while in the other countries of South America, we have multiple biotypes which are specialized to different hosts. So while in the last few years, several studies have been performed with different outcomes, we have a common determinator, that is that we always have some one biotype associated with guava and one biotype that is associated with rose apple and eucalyptus. And sometimes this second biotype, these eucalyptus strains can be grouped in a different genetic cluster. And as it was widely discussed this morning, these exotic strains represent one of the most serious threats to the Australian natural environment and commercial native forests. Based on that, we formed this partnership to try to answer this big question. What are the genetic traits associated with this rapid global dissemination and establishment of the pandemic biotype of Austropuccinia? And we need to do that because first we need to comprehend a disease, a pathogen, in order to be able to manage it. So the first part of this project was conducted in Brazil by an undergrad student called João Morales. So what we did was collect a bunch of isolates of Syzygium jumbos and guava, compared them, and then we found two different isolates that had some morphology differences. We cross-inoculated these isolates in Syzygium jumbos, and we saw that by characterizing the monocycle components, the isolate that came originally from guava was causing a lower density of lesions and severity of diseases. And when we looked these lesions under the microscope, we actually saw that while the isolate that came from rose apple in the first place was able to close its life cycle, the isolate from guava was infecting the plants, as you can see in the bottom picture, forming Austoria, but the life cycle was interrupted and this isolate was not able to sporulate. The same thing happened when we inoculated these two same isolates in guava, but this time only the guava isolate was able to cause the disease. Then we selected these two isolates that have this strong correlation to their original hosts plus one isolate from eucalyptus that's already widely studied to perform the next steps. Before I enter in this next part, I want to bring this little reminder about the rust life cycle that can have up to 5 different types of spores. And we are focusing here in the iridienospores that are the ones causing the disease in the field. So these iridienospores, they are in a eukaryotic stage, meaning they have 2 different nuclei inside each cell, and a total of 36 chromosomes, as Perry presented earlier. So what we did, we collected these iridienospores and extracted the DNA to sequence with PacBio, and we also cross-linked the spores to collect this high-C data from it to be able to phase these 2 different nuclei. We also extracted the RNA of these iridienospores alone or in interaction with its original host to be more accurate in gene prediction, and this data will also be used in further studies that are being developed by the group. All of this went through an extensive bioinformatic approach, and that's what we did in the past 4 months. And we ended up with 3 phased chromosome level assembles. So what does that mean? It means that for each isolate that came from guava, roseapple, or eucalyptus, we were able to phase and assemble the 2 nuclei that have different numbers of chromosomes, as you may see inside the darkest circles. So these 2 nuclei we are calling here as A and B. And this composed assemble, the combination of the 2 nuclei for all the strains, has around 2 giga size, and most of it is composed by transposable elements. We also assembled these mitochondrial genomes, and along with the mitochondrial genome of the eucalyptus strain that was already available, we are now able to compare these strains that came from the center of origin of the disease with the pandemic strain that's widely distributed. So something interesting here is that these 2 nuclei should have 18 chromosomes inside each one of them. But for 2 of our strains, we found actually 2 copies of one of the chromosome placed on nuclei A. And that's interesting not only for Myrtaceae, but also to try to figure out fundamental questions in fungal biology, as it's not something commonly found. And we are still investigating what's happening here, but we also found the same situation in another rust from previous published data. So one of the first things we were trying to answer with that is if these 4 different strains are the same species or could be potentially different species. And by using different phylogenetic approaches, one very common, the other one using all the mitochondrial genes, we found out that thus far all these strains can be considered the same species. Now, the next step is to do a phylogeny of the whole genomes with a sliding window approach to try to either confirm or deny this hypothesis. As I mentioned earlier, this is a work in progress. So over the past 4 months, we were assembling these genomes. And now I will show you our future directions and what we want to do with these new resources. So they will allow us to answer our very first question, that's try to find these genetic traits associated with the rapid global dissemination and establishment of the pandemic biotype. And we can do that by comparing the transposable element compositions, but also to try to find specific genes or gene regions that are associated with these different strains. These resources are already being used to achieve active progress in biosecurity applications. So later today, Luke will present a poster with a high throughput identification and quantification of airborne fungal pathogen Austropuccinia psidii. And tomorrow, Zenian and Austin will present, give a talk on how to prepare for novel incursions using sexy genes to call lineages in a Myrtaceae pathogen. But as I mentioned, these are big genomes. Thus far, those are the biggest genomes of fungi assembled, and it also represents a huge advance in fungal biology. So we will also use these resources to answer fundamental questions. That one of them that we are working with is which are the genetic traits that facilitate adaptation of rust species to either a monoculture, as Puccinia triticina, that's causing leaf rust, or to a diverse range of host plants, that's the case of Myrtaceae. Thank you for hearing me. If you are interested in know more about researches we are doing in Brazil, you can scan this QR code or find me over the next few days. Thank you.
Wow, thanks, Thaís. That was fantastic, really so interesting. Has anyone got questions? Okay, well, maybe I'll ask you one. Do you think that by this identification of the different numbers in the nuclei of the chromosomes, could we develop some way to identify strains based on this? It's a hard one, I know.
Yeah. I'm not sure, because now we are working towards to try to find specific signatures of these differences in the number of chromosomes. So we found 2 strains with the different numbers and 2 that have regular distribution. If we find something that's very specific to this modification in the genome architecture, maybe, I think so. But I think it's early to say because we really don't know why that's happening.